now coming to the most important organelle, as we discussed in the last video, that is about the nucleus. Now when we talk about the nucleus, it might be of different shapes when we talk about the WBCs, the white blood cells, but when we come to the point of the normal animal cell and the plant cell, over there the nucleus is spherical, no doubt in that. Now the nucleus, when we study about the structure of the nucleus, the outermost membrane, it is the nuclear membrane, just as we have the cell membrane for the cell, the outermost envelope part. So this is the nuclear membrane, and this nuclear membrane, most important feature is there are pores. Okay, so there are pores, you can say, but why these pores are there, we will see that. Apart from that, the entire content inside the nuclear membrane is called the nucleoplasm. Cytoplasm, protoplasm, now comes the nucleoplasm. And this small structure, nucleus itself is a small structure in the huge cytoplasm. Inside the nucleus, another small structure is there, that is the nucleolus. Apart from that, there is a lot of thin fiber-like structures, a meshy network. And this is nothing but the chromatin fibers. Very important thing, that is the chromatin fibers. These chromatin fibers, you can see, they are actually very coiled up, extremely messed up, and whenever we talk about the chromatin fibers, we talk about the genetic material they hold. Okay, so these are the ones which actually have the genes, the functional units which actually determine the entire characteristics of your body, be it the physical, the morphological, the functional, metabolical attributes, everything is controlled by the genes that are present inside the chromatin fibers. And these chromatin fibers, they actually, you can say, when they undergo the stage of cell division, they just coil up, they condense, they become condensed and start packing themselves into structures called chromosomes. And the chromosome structure is somewhat like this. The shapes vary. When we talk about the shape of the chromosomes, they again vary, but let me show you. This full structure is the chromosome. This can be treated as one unit and this one as the other unit. Each such unit is called chromatid. The point where both the chromatids they are attached, it is called centromere. Done. The point where these chromatids they are attached is called the centromere. It is that particular place, the centromere is that particular place where the chromosome gets attached to the spindle fiber during the cell division. Now, talking about these chromatids, now actually they have these genetic material well organized, packed into a complete unit. And the genetic material, you know, generally in our case, the genetic material is the DNA. But when we talk about some viruses, over there, the genetic material is the RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. And as the name suggests, deoxy means one oxygen atom is missing over there in the entire structure. That's where DNA has taken the place. And in fact, because of this combination or arrangement, you can say DNA is the most stable genetic material when it comes to the comparison to the RNA, that is ribonucleic acid. Okay, so this was about the structure of a nucleus. Now when we talk about the nucleolus is responsible for producing more amount of your ribosomes and much more of the enzymes associated with ribosomes. So that's where the role comes. Now talking about, as I said in the earlier part of this video, about the nuclear membrane having pores. Now why these pores are there? Is there anything to come out of this nucleus? Yes, definitely. Something has to come out, but what is that? Now using this genetic material, there is a process called transcription, by the help of which certain messenger RNAs have to be released into the cytoplasm. We call them as the mRNA. Okay, after this transcription process, the mRNA is released directly into the cytoplasm. Details about the transcription and then the mRNA being used by the ribosomes to produce proteins, and that process being the translation, they form the central dogma of life. Okay, the central dogma of protein synthesis, that is the DNA replication, the genetic material DNA replication. transcription into mRNA, messenger RNA, and then translated into a complete set of protein. That protein will be actually the reason by which the gene will be actually expressed. If something we are talking about the expression of genes, then it is through proteins. So that's how the messenger RNA has to come out, and it's by the help of this pores, the nuclear membrane being porous, these things come out. Whatever is required to come out into the cytoplasm for more activities, these things help. So this was the basic idea about the gene, the chromatin fibers, the nucleus, nucleoplasm. Now one thing I would like to add, nucleoplasm is more acidic in comparison to the cytoplasm of the cell. And say thanks to this nuclear membrane which completely isolates it from the cytoplasm, though it is porous, still there is a mechanism, there is a structural mechanism over there which regulates it. Okay, so that's all about the cell organelles. And with reference to the nuclear membrane, I would just like to differentiate again between the eukaryotes and the prokaryotes. Now when we were actually discussing about the eukaryotes and prokaryotes with reference to the ribosomes, again over here the nuclear membrane, depending upon the nuclear membrane, these eukaryotes and prokaryotes are initially differentiated. So this is the first point of difference actually, you can say. If in a cell, a nuclear membrane, a well-defined nucleus, a proper distinct nucleus is present, then that cell is called the eukaryote. And if it is not present, it is just like a small region where the nuclear content or the genetic material is stored over there, then that thing is called nucleoid instead of nucleus, and we find that in the prokaryotes or in a prokaryotic cell. Just for the example, bacterial cell of a bacteria. So that's how we have now covered up this entire cytology part. When we talk about the various differences in plant cell and animal cell, we actually talk about the organelles only, the cell wall being present in the plant cell, centrioles and the centrosome being present only in the animal cell. Then presence, what is the similarity? When we talk about the similarity, most of the similarities do exist, and but when we talk about the differences, these differences are very important to be remembered, because generally these are the questions, most probably you will be asked questions regarding these topics whenever you go for any sort of interview or any sort of viva, any sort of tests where these oral tests are taken, and they are considered as you can say a medium through which they get an fair knowledge about what exactly you have learned. So these are very fundamental aspects, you need to focus on them. If you have any questions, anything which you want to know something extra, or you are expecting something more out of it, you can definitely write those questions in the comment box over there, and we will try to reach you the best way possible. The sooner the better. So stay tuned for more videos. I hope you liked it.